Good morning, FOR 345. I'm here live in the studio, broadcasting from an undetermined location because it's a super secret lecture, but not really. Today's lecture continues on, and today is soil aeration. Of course, we have to have our little bit of comic and have one of the better ones that you can see. This deals with the wetland soils. We visited wetland soils last week. So for all you hunters out there, this is kind of the reverse role. Okay, let's think about soil aeration. And so we think first about the atmosphere. We start with the atmosphere, and it's approximately 78% nitrogen, a relatively inert gas, and about 21% oxygen, and about 0.03% or 0.035% carbon dioxide. These are the three main gases. There are others, but these are the three main gases that we deal with. And since today we're interested in how these gases enter the soil, how they exchange. So this is in the context of that exchange of energy, gas, and water. Now today we focus on the exchange of gases. And so we compare the concentrations of these gases in the atmosphere with that in the soil atmosphere. And what do we find? We find that nitrogen is relatively the same concentration. The partial pressure in the soil is approximately the same as that in the air. But when we look at oxygen, it's lower. And we look at carbon dioxide, it is higher. And the question becomes how much lower or how much higher? And the answer really depends on soil physical properties and soil biology. And so we'll explore that today. Some of the other gases, methane, hydrogen sulfide, some of these other very small concentrations, they're important greenhouse gases. But again, today our focus really is on those that really apply to plants, and we'll be focusing mostly on oxygen and carbon dioxide. So what is a well-aerated soil? You might ask, and I might ask you on the next exam. And if we would define a well-aerated soil, we would talk about a soil where the gas exchange is sufficiently rapid. There's a certain amount, those fluxes, that flux of gas that we talked about earlier, that arrow going from the soil to the atmosphere, that has to be sufficiently large so that certain things happen. And an important part of that is that the oxygen in the soil is replenished. And at the same time, that oxygen is used up, and while that's being used up, carbon dioxide is built up. A lot of respiration, a lot of activities, the roots, all of the biota are producing carbon dioxide. And if that was allowed to build up to great levels, that kind of reduces the biota, their ability to function. In the same way, if oxygen gets too low, the respiration doesn't occur. So a well-aerated soil is one where this process of gas exchange allows sufficient exchange of oxygen and CO2 to prevent the buildup of CO2 and to encourage the continual replenishment of oxygen. So that's a well-aerated soil, very succinct definition. And what about this oxygen in the field? So if we look at soils, what controls the availability, what controls the concentrations of oxygen in the soil? And there are a few properties that we can talk about and think about. Okay? And the most obvious one, soil macroporosity. Okay? And so in macroporosity, we have some generalities we made about texture and structure. And we had sandy soils tend to be very high in terms of macropore space compared to clay soils. And we had some structure, well-structured soils versus poorly structured. So essentially those large pipes that allow for macropores, these large spaces that don't hold water against gravity, this is where the exchange takes place. So soil macroporosity is very important. I don't know if my slide is kind of interesting. I hope yours reads the same. It looks like a Cyrillic texture and structure. It just came back. Okay, the second part of that is water content. So remember, we have these pores. 50% ideally would be filled with air, 50% with water. So if all of these are filled with water, the air-filled porosity is going to be low. The exchange is very slow. The exchange in air-filled pore is about 10,000 times more than the exchange of oxygen in a water-filled pore. So soil water content has a direct impact on this process. And finally, the rate of consumption. So we have plants, we have biota, we have a variety of these organisms that are aerobic. They're consuming oxygen, and their rate of consumption draws this down. Okay? So the greater the rate of consumption, okay, the greater the need to have this process occur, this exchange of oxygen allowed from the atmosphere into the soil. And who could forget the soil organisms? Okay. So what's the mechanism? There are two possibilities, right? There's mass flow, just the mass flow of air movement, and there's diffusion. And of course, by this slide, it kind of implies that the real important mechanism here is diffusion. So what is diffusion? Okay, diffusion is the movement of gases to establish an equilibrium against a gradient. So this slide here depicts the process. We have the white gas in the atmosphere. The whites represent oxygen molecules, and we have the yellows representing CO2. So where's the gradient? So in the atmosphere, the concentration of O2 is much higher than in the soils. And the reverse is true of CO2. We look at CO2, and the concentrations, the partial pressure of CO2, much higher in the soil. So this is a gradient, and so diffusion is the movement of these gases in response to a gradient, an attempt to establish an equilibrium. So the carbon dioxide being higher concentration in the soil, it's being produced by a number of roots and organisms by respiration. That tends to diffuse outward into the atmosphere. And the reverse is true for oxygen. Oxygen is higher concentration in the soil, or sorry, in the atmosphere, and it tends to move inward toward the soil. Of course, now we've got to think about the way this is occurring. This is occurring largely through macropores. Those are the large pipes that don't hold water against gravity. If they are filled during saturation, this doesn't occur. So that effective soil texture and structure becomes extremely important because it has a big factor, it's a large factor in regulating this movement, this gradient. Now that is diffusion. We look about the rates, and again, highlighting the importance of these air-filled pores. If you look at an air-filled pore and you compare that to a water-filled pore, the rate of diffusion is about 10,000-fold different. That's an order of magnitude of 4. So you can see why we really rely on this concept of air-filled pore space. And now we kind of build on last week's work in terms of soil moisture and soil moisture retention. And this directly, these ideas of diffusion, you can paste this right over our soil moisture characteristic curves looking for that proportion of macropore space. And we did those calculations last week in lab. How do we assess soil aeration? So we've defined it. We've kind of looked at the process, and now are there ways to assess it? Quantitative ways to say, yes, we have good aeration, no, we don't have good aeration, or we have poor aeration, conversely. And so one of the, I'll give you a number of them, and they're no particular importance, but just in a numbered list. The first one would be obviously the content of oxygen and other gases. And just measuring the oxygen concentration in the soil would give us some idea of soil aeration. And some of those numbers, we have these rule of thumbs, and these are pretty useful to have in the back of your head, particularly during exams, is that in general, the oxygen concentration greater than 10% for most upland plants, that's the requirement. So if we can actually put a sensor in the soil and measure the oxygen percentage, the partial pressure, we say if it's 10% or above, that's adequate for most upland plants. That's kind of a cutoff, a threshold, a number. We think about good aeration, yes, let's say 10% of oxygen concentration. In the same way, we can look at CO2 concentration. So when the partial pressure, the concentration of CO2 exceeds 10%, then we tend to think of that becomes a very bad number. That implies a toxic situation. So we could either measure the oxygen content, or we could measure the CO2 content, concentration, and utilize that as one measure of soil aeration. Okay. And again, it's at the 10% greater for oxygen. Plants need more than 10%, and if you have more than 10% CO2, that's a negative thing. So that number is reversed in terms of oxygen and CO2. Another way we could do is just look at the air-filled porosity, and we talked about the macropore space. And in general, if we have less than 20% air-filled porosity, total pore space, that would, or less than 10% of soil volume, okay, we would start to say that's kind of a cutoff. So 20% of total pore space or 10% of soil volume, that would start to inhibit plant growth. Then it's a fairly easy measurement to do. We could get that measurement probably right off our soil moisture characteristics. And we did that calculation. What's the macropore volume percentage of a soil? Here's a little slide showing the illustration of this and showing how things diffuse, okay? And think about soil. So we might be interested in the rate of diffusion. Okay? And so we could also measure the oxygen diffusing rate. That's so abbreviated as ODR, the oxygen diffusion rate. And this is a measure of the rate at which oxygen diffuses. These is actually done by platinum electrodes, and you insert these platinum electrodes in the soil, and because of the differential in voltage across those electrodes, we can actually measure the rate of diffusion. And the general numbers are, look at those units. We have, if you're less than about 0.3 micrograms per centimeter square per minute, that's the loss of top growth. Okay, so this diffusion rate has a concentration, micrograms, in area, centimeter squared, and a time, minute. So this is a diffusion. It's a rate, amount per unit area per unit time. And so these numbers, 0.3 and 0.2, these are some threshold values. So we can measure this diffusion rate, put these platinum electrodes in the soil, and if it registers less than 0.3, around 0.3 to 0.2 micrograms per centimeter squared per minute, we would say that was a poor diffusion rate. Okay? And so we'd say soil aeration is perhaps less than desirable. We can get these measurements. So you go to wetlands or saturated soils, and sometimes you have very low diffusion rates. And this slide here depicts diffusion of oxygen. Here's some data from a colleague in Georgia. A couple of his grad students, Scott Toriano, that's one of Larry Morris' students, looked at the percent oxygen, percent growth, percent change in oxygen diffusion rate. So here's this grams times 10 to the minus 8 centimeters squared per minute. Okay? And this is 10, 100, 1,000. Okay? So you can relate back to our last slide. If I just go back that way, okay, you see that 0.3 micrograms. Okay, that's 10 to the minus 6, 0.3. Okay. And so where is that here? 0.3 times 10 to the minus 6. So that would be 30 times 10 to the minus 8. Okay. And so we're looking right around here as that cutoff point. Okay. So that kind of relates this. And note the scale. This is in 10 to the grams, 10 to the minus 8 grams. Okay. And if we look at what happens with loblolly pine seedling emergence, so what Scott Toriano did was adjusted the moisture level and measured the oxygen diffusion rate and planted loblolly pines. And those loblolly pines seedlings didn't emerge, okay, didn't begin to emerge until we get around to that critical 0.2 to 0.3 or 20 to 30. Okay, so you can see that the percentage of emerging seedlings is very low until we hit that threshold, and then it increased linearly. So here's a nice illustration of the impact of aeration measured by oxygen diffusion rate on the emergence of loblolly pine seedlings, okay, from 0% emergence to 100% up when we get up around 100. Okay. Pretty nice piece of work. Now, you not only did loblolly pine seedlings, but they also looked at cotton. And it's interesting, so they had cotton roots they measured, and then they measured the biomass in these various conditions. And it was the same kind of a relationship. There was a direct increase of root biomass with oxygen diffusion rate, showing the importance of aeration. And again, the threshold, the cutoff, pretty close to what the loblolly pine was. So very different species, a different system. And here we're measuring seedling emergence. Here they measured root biomass, but the same kind of a slope. So this would suggest that this oxygen diffusion rate is perhaps a useful measurement to assess soil aeration. And these data from two different systems would support that. So we've had porosity, macrofilled pore space, oxygen diffusion rate, and now we look at something else. Another way that we might assess soil aeration would be something called redox potential. And the symbol for that is capital E small h. Okay? And it's measured in volts or millivolts. And so what is redox potential? We can define redox potential as the tendency of a system to reduce or oxidize. So what is reduction? So remember, we define reduction as the gain of electrons and oxidation as the loss of electrons. Okay? So we can measure that, the tendency of a system to gain electrons or to lose electrons. And for example, oxygen, the great electron acceptor, that accepts electrons, okay, and is reduced. Okay, so oxygen is something that we use to oxidize, but electrons flow to oxygen. And so in this process, oxygen itself is reduced. So you can't separate out oxidation and reduction. If something is oxidized, something else has to be reduced. Electrons just don't disappear. They go somewhere. They're either gained or lost. What happens when oxygen is not available? Okay, those, we talked about anaerobic systems. Dissolved oxygen is not present. Then there are other substances that take on these electrons. So the biota continue to try to generate, to respire, then respiration shut down, and so anaerobic respiration, some other processes occur, and electrons build up. And so the electrons no longer flood oxygen. Oxygen is no longer there. So where do these electrons go? Well, the question is first, where do they come from? So you remember glucose, that 6-carbon sugar that everyone knows and studied probably back in cell phys and cell bio. Glucose gets converted in respiration to pyruvic acid and H+, and in the process generates electrons. So this process of generating energy, okay, and respiration generates electrons. And so these electrons have to go somewhere. They just can't disappear in the system. And of course, they go to other things. So if oxygen's not around, they go to iron. So here's a nice illustration showing this measurement of redox potential. Here's EH on the y-axis, and here it's measured in volts. And note the scale. We're going in tenths of volts, okay? And 0 positive, and we go negative, and then minus 0.2, minus 0.4, minus 0.6. And we look at days after saturation. So essentially, what happens after a rainfall? Okay? And so here's the measurement of iron oxidation versus reduction, and here is EH. So after a rainfall, under very saturated, we start after saturation, we start with the EH drops precipitously. Okay. Okay. So what happens after rain? After rain, all the plants are looking for oxygen, and everything's stopped. All those pores are filled with water. Okay. So immediately, the process of respiration generates all these electrons. They've got no place to go. And the system goes from a very positive EH to a very negative EH. Okay? So it's a very precipitous drop. The same thing occurs. Look at dissolved oxygen. Dissolved oxygen drops. Here's dissolved oxygen on the right. Okay. Dissolved oxygen drops precipitously along with EH. And what happens with pH? We'll discuss pH next week. pH, the hydrogen ion concentration negative logarithm. But during the system, when that happens, the pH increases. So a very dynamic system, as EH is going down and dissolved oxygen going down, pH is going up. And so it creates a very complex relationship of electrons being generated, electrons moving, being oxidized and reduced. Let's go back to our EH. So we go down the EH, we've got the system, it's rapidly, all the dissolved oxygen used up. And then as the system begins to drain, okay, 6 or 7 days, here we start to see a great improvement in the situation. Okay, the soil drains and the EH climbs back up. So let's look at the relationship between EH and oxygen diffusion rate. And here's a peat bog. So here we have oxygen diffusion rate, okay, 0.12, and it's micrograms per centimeter squared per minute. And remember, 0.3 and 0.2, those are the cutoffs for pore aeration. So as the oxygen diffusion rate becomes higher, okay, the rate becomes greater, the EH becomes greater. So you start to see a relationship between these variables that we've used to assess aeration. And they're correlated. As EH increases, oxygen diffusion rate increases. As EH increases, dissolved oxygen increases. As EH increases, the macroporosity increases. So all these numbers, they're related. And we could maybe use one or another of them to assess soil aeration. Any one of them might do. Some are more difficult to measure. The platinum electrodes for oxygen diffusion rate are very, very finicky and difficult, and that's a tough measurement. But the probe for oxygen and CO2 is very simple, and that's a very easy measurement to get. Let's look at the process. So we have, we have iron here, the +2 valence, and it's going to +3, okay? So what's this process called? We go from a +2 to a +3. We lose electrons, okay? Okay, that is oxidation, okay? And in the process, we generate electrons. Those electrons have to go somewhere. So where do those electrons go? Okay, they don't just disappear. Okay. And here's an example of what happens. Oxygen accepts the electron. So we have this oxygen, this half mole of oxygen, with hydrogen and accepts this electrons, and now we have water. So oxygen itself is reduced to water. Okay, so iron, FeO plus 2 going to Fe plus 3, that's oxidized. So that oxidation is due because the electrons flow to oxygen. And then we make water. And of course, water is the reduced form of oxygen. So it's a fairly linked system. One thing occurs not in the absence of the others. Oxidation reduction is a paired reaction. So when you see that oxidation of iron, you see it written this way in the textbook. And we talked about how iron is oxidized, and we talked about weathering. I think we use this example, FeO going to FeOOH. Okay? So this we didn't show all those electron balances, the electron transfer. But now we show that explicitly to make the point that the electrons that are lost from iron go somewhere and they flow to oxygen. Okay? So in the presence of oxygen, iron is oxidized. That brings us into another way to evaluate these aeration status, and we can think about trying to look at reduction. And so actually Marty Ravenhorst and some colleagues down in Maryland devised a very interesting tube called the IRIS tube. And IRIS is short for Indicator of Reduction in Soils. So these are tubes that are actually painted, PVC tubes painted with an iron compound. And here they are. So this iron compound is yellow. Okay? And these tubes, these are from we had an experiment out at near the Seneca Falls, Montezuma, and looked at reduction. So these were placed in a wetland. And what happens is that when you put these tubes in the ground, this iron in this paint is reduced, and reduced iron turns gray. And so we can actually quantify the area. Here's some orange iron, and then gray. So you can actually use this to quantify and demonstrate reduction simply by a change in a paint pigment of a buried tube. You put these tubes in for a couple of weeks and pull them out, and it's a nice way to demonstrate reduction. Here's a picture of that in Montezuma, these installations. This was a field trip we had last year. Very pronounced wetlands, continually saturated. Place these tubes in. And so now we've got the picture. Let's think about what's actually happening. So here's a table from the text on page 273 on oxidized and reduced forms of elements. It kind of gives some little more feel, a little more understanding for what's going on. And this shows two things. It shows the oxidized form. Here's oxygen. Then it shows the reduced form, water. Now we just demonstrated that. We just showed how oxygen gains electrons and is reduced to water. Okay. Now we can also measure the potential. Okay. We can measure the EH at what that happens. And that change of oxygen to water takes place at an EH of 0.38 to 0.32 volts. Okay. 0.38 to 0.32 volts. Now these are volts. If we had measured that in millivolts, it would have been 1,000 times that, or 380 to 320. Okay. So sometimes you'll see this measured in millivolts. But oxidized, reduced, that takes place. The EH is somewhere at 3832. Now what happens? What happens when all this oxygen runs out? It's depleted. The roots have used it all up. There are other things. And at that point, there are other materials, other elements that accept electrons. So after all the oxygen is depleted, nitrate, NO3 minus, accepts electrons. It's reduced to N2, gaseous nitrogen. Here's a process, okay, called denitrification we'll talk about later. But this is a very important function of wetlands. Okay, nitrate in large concentrations is toxic. It's a plant nutrient. But this is one way that nitrogen is removed from the biological systems in wetlands, and it turns to gas. Okay. So this takes place at an EH, lower EH than O2. So this takes place 0.28 to 0.22. So suppose then we have these processes going on and we utilize all the nitrate, convert that all to N2. What happens next? The next thing that accepts electrons is manganese. So manganese is reduced, goes to -2, okay, and that's in the EH of 0.22 to 0.18. Eventually, all the manganese is reduced, and when that happens, iron plus 3 goes to iron plus 2, okay, and that's reduced, 0.11, this the EH range, a lower EH range yet. And now, when all that's done, and the next thing that happens is sulfate. SO4 minus 2 goes to sulfide, S2. And what happens, that's a very negative EH. And when all the sulfates utilize that way, the next thing that happens is carbon dioxide is reduced to methane. So here we are in a very anaerobic system. All these other things have been reduced, and now we're down to carbon dioxide to methane, very negative. So this is very, very, very poorly aerated soil, very negative EH. And it shows the progression of decreasing potential and which of those species of elements accept electrons. So there's an awful lot in that graph. And it kind of encompasses or encapsulates, summarizes all the things we've just talked about. Okay. That's a lot to swallow and probably a good thing to think about. That's an important point. Now, if we're going to use this number, this EH, this potential, to indicate a well-drained soil, what number would we choose? So we look at this chart and try to think about that. We have O2 being accepting electrons, 0.38 to 0.32, then nitrate, then manganese, okay, and so forth and so on, +4 to +2. So we look at that, we think, what number could we use to characterize a well-drained soil? Okay. Well, it certainly wouldn't be a negative number. Okay. And so the answer to that, somewhere between 0.4 and 0.7 volts. So here's our next criteria. So we had an ODR of 0.2 to 0.3 micrograms per minute per centimeter square per minute. We had a 10% oxygen concentration and so forth. And now this number, this threshold using EH, is somewhere at a 0.4, 0.7. And given what we just observed here, that's just above where all the oxygen begins to be depleted, this is a reasonable number. So again, this is a useful number to know, the threshold for a well-drained soil for an EH value. Okay. So now we've measured it. We've kind of given some different numbers to indicate aeration status, and now we might go a little further. It's probably useful for us to think about the factors that affect soil aeration if we wanted to predict soil aeration. And then we could actually link this back to our soil physical properties and our water. And again, we're building on that first idea, of this exchange of energy, water, and gas constrain the biota. And now we'll look at those soil physical factors that we think about to be able to predict aeration. Okay? Right back to the beginning of the course. And the first and most obvious, macropore space. We've seen the importance of diffusion, and that diffusion doesn't take place in water-filled pores. So we talk about macropore space. And so then we say, okay, what are the physical factors? What are the indicators? How do we measure or quantify macropore space? And one of those is texture. And we talked about generalizations of texture in macropore space, with sands being fairly high macropores relative in terms of their total pore space, and some of the finer textured soils being small. And then we talked about structure, as structure modifying the effect of texture. So again, this pipe system, going back to the original idea of the soil physical properties, very, very important. And we add on to that, we also, what are the things that contribute to aggregate stability? Okay? And so bulk density and organic matter, and we talked about structure. So all those things affect macropore space. And recall also that we were thinking about depth in the profile. We talked about how the exchanges of energy, water, and gas are maximum to the surface and decrease with depth. So as we go deeper in the profile, that macropore space is reduced, and we talked extensively about that when we talked about bulk density and structure. So macropore space then is one of those prime factors that we think about for aeration. And then we could finally relate this back to our parent materials, the lacustrine materials, the outwash materials, and so forth. The other thing is the rate of consumption of oxygen and the rate of production of CO2, so the biochemical reaction rates. And this should make sense. The microbial decomposition of organic residues, the biota, they churn, they chew up the stuff, and in doing so, they generate a lot of carbon dioxide. They're also utilizing oxygen for energy. So the greater that rate, the greater the need to remove that CO2 and to input oxygen. Okay. Roots are one of the more important organisms. Okay, we think about plants, but those roots, they are very, very active. They're generating a lot of CO2. They're producing a lot of carbon dioxide. Then there are a whole slew of biota, of fungi and microbes associated with root systems in the rhizosphere. And these are doing the same thing. They're utilizing oxygen for respiration. They're generating CO2 from that respiration. So this whole biota system is utilizing these oxygen gases and generate a lot of CO2. And this exchange is critical to their continued production. Season. So season kind of goes along with both of those. We have seasonal impacts on macropore space and biochemical reaction rates. And one of the nicest graphs that illustrates that is in the text with corn. Okay? So here's a very nice illustration. So here's a cornfield in Missouri, and we go from June to November. So we essentially the growing season, then the post-growing season or the fall, and soil depth. So we're looking at the combined effect of soil depth and season, and this is showing carbon dioxide concentrations. So we start out in June, after planting, when the plants are just getting established, and we see the carbon dioxide concentrations 0.3 to 0.5, okay, relatively high compared to a forest soil, but low in an ag system, and they are increasing with depth. Okay, this is an expectation you should be comfortable with. There's less exchange, okay, most of the rooting activity is at the surface, but roots are developing over time. So as the growing season proceeds, those carbon dioxide concentrations increase tremendously. Okay, look at there, more than 7%. This is a tremendously high concentration in the middle of the growing season. The biochemical activity is phenomenal. The roots are just sucking the stuff up. The plants are growing. They're putting out the stalks and fruiting. It's just a very intense time of biological activity. As we go toward the end of that growing season, the carbon dioxide's lower a little bit. Okay, and they have an irregular pattern. They increase with depth and decrease. Okay, and they're increasing. This depth is the maximum part of the root zone, this very intense part of the root zone. Okay. And then as we go into the fall, the levels dissipate further, and we go back to more of a situation what looks like in the spring or the early summer. So this is a very nice graph showing the interaction of concentration with season and depth. Okay, and it's just a wonderful illustration of also the very high concentrations that can be attained. So you know what corn looks like. It's planted very densely, okay, the root systems, and these concentrations are very, very high. Okay? And you'll see none over 10%. Remember, 10% is that cutoff where aeration ceases. If they get over 10%, your corn's probably not going to be looking too good. One question you might consider, I won't answer it here, but I'm going to ask you 7%, how much more is this than atmospheric? Okay, so what's the relative increase as we go from the atmospheric concentration to the soil, and we're looking at 7%. How many times greater is the concentration of 7% compared to the atmospheric? So to answer that question, you'll have to know what the atmospheric carbon dioxide concentration is, and then you have to divide. So I'll leave that to you, and that's, we'll come back to that question again, probably on an exam. Let's contrast that. We have forest soil, so that's a very intense ag system. Let's look at a forest soil. Okay? So here's some data from Bear Brook Watershed in Maine. So this is some data from Ivan Fernandez from one of our gradient studies when I was working up there at the University of Maine. And we had studied the concentration of carbon. We were looking at exchange rates in a variety of systems, cost of climate gradient, all the way from the northern part of the state to the southern part. So here we show the soil air concentration, CO2 concentration, microliters per liter, okay, or you know that as 10 to the minus 6, okay, parts per million. And we go from 0 to 5,000 parts per million. And we also show it by horizon. So we have the O horizon, the B horizon, and the C horizon. And we go season. We go from January all the way to December, an entire year. And a couple of patterns are interesting here. Okay? And the first pattern is that the carbon dioxide concentrations are maximum, okay, just about the middle toward the end of the summer. This is in a spruce fir stand. Okay? And the other thing is that the patterns with depth change over time. So let's look first at the O horizon. Okay? So the carbon concentrations in the O horizon are the lowest. Okay? And of course that makes sense. That's the closest to the surface. And they peak a little earlier in the season. We go down to the B horizon. The concentrations are higher. Okay? This shows increasing concentration with depth. And they peak just a little bit later. And finally in the C horizon, the concentrations are higher yet, and they peak later with depth. So the rates of exchange kind of lag, and so the exchange with carbon dioxide and oxygen are lowest in the C horizon and highest in the O horizon. Okay, so look at these numbers, 1,000, 2,000, 3,000, 4,000, 5,000 parts per million. Now I'm going to ask you to convert that to percent. Okay, so here we have a forest soil, and compare that to an agricultural soil. So I'm wondering, my question to you to think about, is these peak concentrations, 4,000 parts per million or 4,000 microgram per liter, what is that in percent and how does that compare to an ag system? Okay? So again, I'm going to leave that to you and I'm going to ask you to convert that, and when I see you again in person, I'll ask you for those numbers. And that begs the next question. In this system, how much more is this in the soil relative to what's in the air? What's the factor of increase? So 3 questions for you to answer when I see you next time. Okay, we've talked about it, we've quantified it, we've looked at ag systems, we've looked at forest systems, we have some pretty good data. What are the impacts of poor aeration? Well, the first and obvious one, reduced root growth. And we think back to that graph with the oxygen diffusion rate in that 0.2 times 10 to the minus 6 microgram or centimeter square per second. Okay? That's clearly reduced root growth, and when you have those high concentrations, the roots cannot respire and they stop growing, and sometimes they can even be killed. Okay? So it's not only growth, but roots have a function, and their primary function, aside from stability, is to uptake nutrients and water. So as the CO2 concentration increases, the O2 decreases, as aeration becomes more poor, root nutrient water uptake cease. And so that's a direct result. Now these are related because that of course impacts growth. The microbial decomposition rates slow down. Last week we visited organic soils, and those organic soils were there because the rates of accumulation far exceeded the rates of decomposition. And those are in wetlands. Of course, in wetlands, the reduced oxygen levels are very, very low, okay, practically nil. They're anaerobic conditions, so material doesn't decompose, it builds up. That is a negative impact. This microbial decomposition rate in upland systems is pretty important because it's responsible for cycling of nutrients. Another consequence is buildup of toxic materials. Some of these gases, methane, hydrogen sulfide, these are toxic. CO2 is toxic, and those materials build up and they contribute to the inability of roots and other organisms to function. Here's a nice slide depicting active negative impacts on poor aeration. So how many times do you see this in a development? Okay, so here's a tree, the homeowner desires a tree, brand new house, and all around the street there's no protection of the soil. So immediate first thing is soil compaction, okay, and increasing in the density. But the associated problem with that is the decrease in macropore space and a subsequent decrease in aeration. And this is often the death knell for the tree. There are a whole lot of species that just cannot handle this. So 2 or 3 years after the developer disappears with your money, you've got a $5,000 bill to remove this tree between power lines, and it costs anywhere from $1,000 to $5,000 to have this done, depending on the tree size. And if you don't take care of it, of course, the next thing you have is a lawsuit when it falls on someone else or someone else's house. So what are some management alternatives? What can we do when we have poor aeration? What are some solutions to this possible potential growth limitation? Well, improve drainage, and we talked about this. Recall our soil water lab. We talked about the effects of ditches and drainage tiles and raising beds. By doing that, you can get the aerated zone thickened or increase the aerated zone. And we showed how to space drainage ditches, and we talked about this in drainage tiles, and we actually looked at this in our organic soils lab. We saw the various drainage tiles and ditches and saw how low the water tables were. So that's one way to do it. This may be practical on large areas. If you're growing vegetables, it's practical. It used to be a very practical way to increase the growth of southern pines in the coastal areas. A system of drainage ditches made a tremendous difference in the growth of pine from non-commercial to commercial. So a lot of drainage ditches were installed in the southeast in the 60s and 70s. Another way to do it is to just accept poor drainage or poor aeration and utilize the appropriate species. There are a number of species that are able to effectively compete in poor aeration, black spruce, for example, and we've talked about some of these in the past. Red maple can handle some pretty wet conditions. What are the species that can't? And think about our trip to Three Rivers, our first field lab in terms of soil morphology, and the red pine. And they were doing extremely well in that very well-drained, somewhat excessively drained Otisville soil. And then next week we visited Heiberg Forest. We went down to a Volusia Chippewa system and looked at red pine, and essentially it was dead pine. So just simply managing the correct species is a very important piece of this, and that's one thing that ties the utilization of soil information to land management. In large land pieces, it's really often not available, the resources aren't available to be able to do these massive changes of aeration, and so then we're kind of dealt the hand, and you've got to manage the hand you're dealt with, and that's by managing the appropriate species. Another way to improve aeration is with aeration tubes. So a lot of times in the horticulture industry, you see these tubes with holes sticking out. So these are inserted in soil to actually improve root aeration. Okay? And of course, in the horticulture industry, there's a whole other set of resources available. These trees, individual trees in individual plantings, worth thousands of dollars in landscape context. And so the ability to treat or to manage the situation or to manage the site is much greater than you have in a forest situation. Okay? There's a lot more at stake, and there's a lot more capital available to improve the situation. In lawns, there's a whole operation, ChemLawn, and other companies that make their living in making lawns lush and green. And one of those involves soil aeration. Here's a treatment that actually takes little plugs of soil out. Now, it doesn't compress the soil. It takes little plugs out and provides little holes. So you see this a lot on golf courses, these almost like little tiny divots. And these holes allow proper aeration to some of these lawns. So it's not all bad. There are some very positive impacts of poor aeration, and we actually utilize poor aeration systems, and we visited some of these systems last week. We visited wetlands. We utilized the advantages of poor aeration. Okay? And one of those is wetlands. And so what is a wetland? So we define a wetland as soils that are water saturated near the surface for prolonged periods. And there's a key thing here. This little piece, temperature greater than 5 degrees C. This is a key component. Saturation before you get to the growing season doesn't make any difference. But once you have organisms trying to subsist, once you get to this temperature greater than 5 degrees C, then it becomes important for aeration for a lot of these organisms. So the wetland definition, water saturated near the surface, prolonged periods, and a temperature. It's kind of a fuzzy definition. It says, well, and so that's led to a lot of problems and a lot of refinement with what wetlands are. So what are wetland characteristics? Okay, so we have to first define a wetland. So we need this is a little bit of a fuzzy definition. So we say, well, we need a little more than that to really tell what a wetland is. And so we define wetlands based on 3 criteria: the hydrology, the saturation, hydric soils, okay, the presence of very wet soils, poorly drained soils, and hydrophytic vegetation. So the Army Corps of Engineers utilizes these 3 criteria to define a wetland. And this is the beginning. These are the criteria, and there are several conditions to meet each of these. So when two of these 3 are present, the Corps, the Army Corps that regulates and is in control of the Clean Water Act, you have a wetland. And so many of you, when you get out of here, your first couple years at a consulting firm, environmental consulting firm, actually might be doing some of these wetland determinations, wetland delineations. So let's look at the piece of those. Let's first look at hydric soils. Okay, hydric soil. So again, we start with a definition, and a definition that maybe is a little bit loose and fuzzy, and because of that has some indicators. But let's look at the definition of hydric soil. Hydric soils are formed under conditions of saturation, flooding, or ponding. Okay, so they're wet. Saturation, flooding, or ponding. And then when? Okay, it's long enough during the growing season. Okay, we come back to the same idea, the growing season, temperature greater than 5 degrees C, soil temperature. And long enough, and the result, anaerobic conditions in the upper part. Okay? So conditions of saturation, flooding, or ponding, long enough to develop anaerobic conditions in the upper part. And now we say, if you're a lawyer, you're starting to say, well, what's this upper part? and how anaerobic, and long enough. So this leads to some questions, and if you were a lawyer, you'd have somebody up the stand sweating, but we can solve that with some indicators, and so we go on. Okay, so saturated soils, these are saturated and reduced, a little more definitions, a little more compelling, saturated and reduced. Okay. And so we come to this need, well, I still need more specifics. I'm still not very certain I know what a hydric soil is. And so then we revert to hydric soil field indicators. Okay? And so these are indicators, these are morphological indicators associated with reduction. And there's lists and lists of these. And here's one, loss or accumulation of iron, sulfur, manganese, and carbon. So what happens in wetland systems? Okay? Iron and sulfur and manganese and carbon, these are accumulated. Okay? Here's a depiction. Here we show decreasing period of saturation. This comes from the text. Okay? And here we have hydric soil. So we have a couple of indicators. So organic matter, a soil feature. And we have paraqueic and aqueic, these very wet moisture regimes. Okay? And organic matter is very high in hydric soils. Okay? And this Gley chroma, chroma less than 1. We talked about reduced conditions in less than 2, chroma less than 2. So Gley, even grayer, less than 1. So on less than 1 on that, remember on the lightness chart and the chroma chart? When you have over in those left-hand columns a very low chroma, those are hydric soils. Okay? Less than 2 in concentrations. So concentrations are small, almost like little pellets, small round, almost like pellets. And that's manganese, and that gets reduced. So when you have chroma less than 2 in concentrations, those are hydric soils. Okay? Chroma greater than 4, nope, too bright. And the other one is concretions, nodules and concretions. So concretions are these very hard concentrations. You can actually, they're self-formed and they're manganese. And so when you have a lot of those are hydric soils. Those are hydric soil indicators. Okay? They help indicate the presence of a hydric soil. It's a little bit more fulfilling than that definition, long enough and wet enough. Okay? Here we go back to our color chart. And remember, low chroma. So low chroma, we're looking on these, here's chroma 1, okay, for 10R, 7.5YR, and 5Y. Okay, and we're looking for these low chroma, these gray colors. Here's gray with a lot of red in it. Okay, low chroma, less than 1. Those are wetland hydric soil indicators. Here's an example. This is low chroma. This is also modeling. Okay, this is chroma 2 in modeling. Okay, that's a hydric soil indicator. Okay. Here's the very low chroma, 1, and here are oxidized root channels. Okay, so this is another indicating of a higher hydric soil, low chroma and oxidized root channels. Okay, this root channel staining. So the root oxidizes iron just around the edge, and everything else is reduced. Of course, that root's pumping iron down here. Sorry, pumping oxygen down. Okay. Here's an illustration with depth and EH. Okay. So here we have water, and as we get the EH decreases, okay, you get this little aerobic zone, transition zone, then you get into the saturated zone. You can see the EH take a dive. So here's a nice indication of the effects of saturated on EH, on depth, in the anaerobic zone. And here's an illustration of that root channel. Okay, the water comes down, but in that root channel, iron is actually oxidized, and so the EH is a little bit higher. And you can look at this up. There's a nice booklet published by the USDA, Field Indicators of Hydric Soils in the United States. And all of these and many others are in there. And this is what soil scientists use. So if you go out with the Corps of Engineers and on the soil science detail, you'll be utilizing these indicators to define hydric soils, which are part of this wetland. Of course, you'll have another person specialist doing the vegetation, or by then you may have learned all the vegetation, the facultative anaerobes and all the other vegetation that qualify things for hydric soil. And that's the redneck version. There's sandy and glade and a depleted matrix. Here's the removal of iron and depletion. It's reduced iron. And I guess that's a good place for us to stop. I hope we've digested some of this. We will continue on, and when I get back, we'll talk about soil temperature and we'll finish up soil moisture regimes. Hope that you enjoyed the exam and did well, and I will get those graded over this weekend. And I will see you Monday. Have a great weekend and think soil.